Hello, greetings to all of you. My name is Muhammad Faris, and my fellow members will be presenting to you a journal review paper by Kabbalah and Turan in 2026 about the DNA barcoding in forensic entomology, a molecular tool for reliable postmortem interval PMI estimation. So, without further ado, let's get started. So these are my fellow group members that will be presenting with me. Muhammad Faris bin Ahmad Ruslan, which is me, Muhammad Mukri Hazib bin Muhammad Yusuf, Nura Atikah binti Muhammad Azib, and the last one is Bil Istina Alia binti Muhammad Razib. So, welcome everyone. Today, we are discussing a vital aspects of modern criminal investigation, forensic entomology, the study of insects to help solve crimes. When investigators find insects on or near a body, these tiny clues can tell us a lot about the time of death or postmortem interval PMI. This is especially crucial when the body is in advanced stages of decay, where traditional medical methods might not be enough. However, the success of this method hinges entirely on knowing exactly which insect species we are looking at. Traditionally, this meant relying on physical identification, looking at features like shape or size. But as noted by Kabbalah and Turan 2026, this is not always reliable. Sometimes, the experts needed aren't available on site, and often, the insects themselves are too damaged or too young to identify by sight alone. To solve this, we now use a method called DNA barcoding. This involves taking a small standardized piece of the insect's DNA, specifically from the cytochrome C oxidase I (COI) gene, and matching it against the global database like BOLD or GenBank. It's like using a digital fingerprint to quickly and accurately name the species. Next is the forensic bottleneck or limitations. So why do we need this molecular approach? We run into what experts call a forensic bottleneck. First, we face morphological barriers. Insects found at the scene are often eggs and very young larvae. They simply haven't developed the physical features needed for identification. Second, there is the cryptic species problem. Some species look identical to the naked eye, but actually grow at a very different rates. If we misidentify them, we miscalculate their growth, which can shift the timeline of death by days or even weeks. And finally, there is a sample degradation. Crime scenes are messy. Specimens are often damaged or fragmented. As Kabbalah and Turan 2026 emphasize, these failures are high risk. Misidentification doesn't just mean a wrong name. It introduces profound errors that can compromise the integrity of the entire investigations. Next is the paradigm shift, molecular identification via DNA barcoding. This brings us to the ultimate solution, a major shift toward molecular identification using barcoding. What makes this unique is that it moves forensic entomology away from subjective visual guesswork, turns it into objective data-driven science. It works because of a rule called the barcode gap principle. In simple terms, the genetic differences between different species are much larger than the minor genetic variations found within a single species. This creates a clear gap that lets us tell them apart perfectly. This also gives us massive systemic advantages. DNA barcoding is rapid, repeatable, and standardized. It completely bypasses the old issues because it doesn't care if the insect is an egg, a larva, or a crushed fragment. To do this, we target a highly specific region of the DNA, a standardized 658 base pair region of the mitochondrial COI gene. By focusing on this exact genetic target, we ensure that every identification is accurate, reliable, and strong enough to hold up in the court of law.
Forensic entomology often deals with damaged specimens, egg, or larvae that are difficult to identify morphologically. DNA barcoding using the COI gene helps achieve highly accurate species identifications, including cryptic species that look similar but develop at different rates. Flies are important because they are the first insect to colonize a body. Studies show that even degraded DNA can still be identified using short mini barcodes. Clear barcode gaps between species also make the analysis reliable for forensic investigations. Beetles are mainly useful in late stage of decomposition and skeletal remains. Some species are active during colder seasons, making them important for winter PMI estimation. Different beetle groups also appear at different decomposition stages. Accurate species identification improves PMI estimation because misidentification can affect the estimated time of death. Genetic analysis of insect can also help to determine whether a body was moved from another location or environment. For species confirmation, forensic scientists compare DNA sequences with databases, such as BOL and GenBank. BOL is preferred in forensics due to its detailed specimen records, while GenBank provides broader sequence data. Cross-checking both improve reliability.
Hi everyone, my name is Muhammad Mukriazik bin Muhammad Yusuf, and right now we are going to look at the alternative in order to solve modern forensic challenges. So the first alternative is mini barcoding. So our main problem is that DNA in forensic sample is often degraded by heat, UV, or chemicals. So the solution is to use mini barcodes, a short fragments between 100 to 300 pp. The success rate is it is high accuracy even with historical museum samples or old evidence. The key finding is that a 130 BP fragment is just as accurate as the full 650 BP sequence. Next, alternative number two is next generation sequencing and meta barcoding. The main problem is that traditional sequencing is too slow and is expensive for hundreds of larvae. So the solution is next generation sequencing. For a massive scale, this technique allows us for bulk sampling when analyzing hundreds of insects in a single run. And the efficiency is that it can detect over 30 different species at once, including rare ones. And for bonus, is that it is fast and low-cost tools like PCR, HRM are now emerging for quick ID. So last but not least is alternative number three, which is multi-marker approach. The problem is sometimes the COI gene is not enough to separate sibling species, and the solution is to use multi-locus barcoding, where we combine COI with other genes. The other markers include ITS2 or nuclear that are better at distinguishing certain flash flies, or as known as sarcophagidae, and then COII, Site B, and antigens that provides extra layers of verification. So the future hopes is that sequencing the entire mitochondrial genome for maximum data. That's all from me. Thank you everyone.
Which of these three will you choose? Mini barcoding, NGS and meta barcoding, or multi-marker approach? The answer is multi-marker because in forensic entomology, it provides the highest accuracy and reliability in species identification, which is essential for precise postmortem interval (PMI for short) estimation. Multi-marker actually exceed the standard DNA barcoding method, which use the CO1 gene for species identification. In CO1 gene, some closely related or siblings species may share very similar CO1 sequences, making identification difficult and increasing the risk of forensic error. Multi-marker actually solves that by using several markers together, which provide stronger verification and improve the ability to distinguish closely related species accurately. To justify the answer, it has highest accuracy in species identification. In forensic entomology, incorrect insect identification can lead to inaccurate PMI estimation by days or even weeks. The motor marker prevent genetic overlap between species, which make it easier to detect. Next, more reliable for court and forensic use. Forensic evidence must be scientifically strong and defensible in court. It provides multiple layers of genetic evidence, increase confidence in final identification, and reduce chance of false matches. The third effective of cryptic or sibling species in some forensic insect species, as they are morphologically similar and genetically close. Thus, it is useful in difficult forensic cases where precise identification is critical. Lastly, for future potential, the approach also supports future developments such as advanced phylogenetic analysis, AI-assisted species with identification. This means not only useful now, but also adaptable for future forensic technologies.
So for the conclusion, when standard medical timelines fail, the accuracy of murder investigation depends entirely on the biological evidence left behind. As we have shown today, traditional physical identification methods introduce too many risks of human error. Look-alike species, damaged fragments, and young maggots often cause mistakes that can throw off a death timeline by weeks. The clear value of the research by Kabbalah and Turan is that it completely removes this guesswork. By using DNA barcoding and the genetic boundaries of the barcode gap, we replace personal opinion with clear genetic facts. We are no longer guessing based on what an insect looks like under a microscope. We are reading its actual DNA code. Ultimately, this changes forensic entomology from a subjective observation into a highly accurate, data-driven science that stands up perfectly in court of law. It gives investigators a reliable tool to find the truth and ensure the scientific evidence can be fully trusted in the justice system. And that's all from us. This is the reference that we use throughout the presentation. Thank you for your attention. Bye.